Good afternoon. My name is Gillian England-Mason, and I'm a postdoctoral fellow working with the APrON study team under the mentorship of Dr. Deborah Dewey. Today we will be discussing with you the results of our research program, which examines how exposure to plastic chemicals during early development affects children's genes, brains, and behavior. In order to introduce you to this topic, I want you to take a look at a familiar scene. This is an image of a typical Canadian grocery store aisle. The shelves are lined with a variety of colorful plastic food and beverage packages. Food is emerging as one of the most important sources of exposure to harmful plastic chemicals. However, diet is not the only relevant source of exposure to these chemicals. During this presentation, we will discuss the various routes of exposure to plastic chemicals, why exposure to these chemicals in pregnant women and developing fetuses is of growing concern, and the findings from some of our research examining how early exposure to plastic chemicals affects child development. Endocrine disrupting chemicals, or EDCs, are industrial chemicals that can disrupt the activity of hormones and the body's normal functions. Two main classes of EDCs include bisphenols and phthalates. Bisphenols and phthalates are mostly man-made plastics and plasticizers. They are found in a variety of consumer goods, such as food and beverage containers, textile and building materials, personal care products, and children's toys. Bisphenols, such as bisphenol A or BPA and bisphenol S or BPS, are commonly found in the lining of canned goods, hard plastic containers and toys, and thermal paper receipts, whereas levels of certain phthalate metabolites, such as MEHP and MEHHP, come from exposure to vinyl building materials and flexible food packaging, while levels of other phthalate metabolites, such as MBP, come from exposure to the solvents used in personal care products, such as soap and nail polish. Humans are directly exposed to these plastic chemicals through the ingestion of food and beverages, the inhalation of particles in the air, such as dust, and absorption through the skin. Given the ubiquity of these plastic chemicals in daily life, over 90% of the Canadian population is continuously exposed. So why are we interested in the effects of EDCs? Exposure to EDCs contributes to disease and incurs high economic costs. The cost in terms of adverse health effects associated with exposure is estimated to be $340 billion in the USA, which amounts to approximately 2% of their GDP, and $217 billion in the European Union, which amounts to approximately 1% of their GDP. Over the last few decades, exposure to EDCs, such as bisphenols and phthalates, has been found to be associated with altered reproductive function in both men and women, increased incidence of chronic health conditions such as diabetes, respiratory diseases, and cancer, and abnormal growth patterns and neurodevelopmental delays in children. Although there is a variety of associated health effects, the greatest proportion of the overall cost of exposure to EDCs is attributable to neurological conditions and neurodevelopmental difficulties. One of the important issues when it comes to the effects of EDCs is considering the age at exposure. In addition to the direct routes of exposure, bisphenols and phthalates can be indirectly transferred from a pregnant woman to a developing fetus through the placenta, or from a mother to a child through breast milk. The World Health Organization states that pregnant mothers and children are the most vulnerable populations to be affected by these environmental exposures, and that the effects of early exposure to plastic chemicals may not become evident until later in life. Accumulating data suggests that many of the health conditions associated with exposure to these chemicals have fetal origins, but the causes remain unexplained. Gaining a better understanding of how prenatal exposure to plastic chemicals impacts development will help to improve public health and the well-being of society. In order to address the mounting concern over the effects of plastic chemicals, and as part of the APrON study, our research team examines how early exposure to plastic chemicals, specifically bisphenols and phthalates, affects child neurodevelopment. Today, we will briefly discuss some of the findings from our broader program of research. First, we will go over two of our studies which examine how prenatal exposure to plastic chemicals is related to brain structure in young children. Second, we will discuss some of our completed and ongoing projects which examine how prenatal exposure to these chemicals is related to behavior and cognitive development in young children. And third, we will briefly touch upon emerging research which suggests that prenatal exposure to these chemicals may alter the epigenome and that these epigenetic alterations could underlie the neurodevelopmental difficulties we see in young children. Based on research using animal models, we know that in utero exposure to these plastic chemicals affects the brains and behaviors of rats and mice. Further, from observational research in humans, we have seen evidence that prenatal exposure to these plastic chemicals is associated with behavior problems in children. This includes both internalizing behaviors, such as anxiety, depression, and withdrawal, and externalizing behaviors, such as attention problems and aggression. So in 2 of our studies, we have used advanced neuroimaging techniques to examine the associations between prenatal exposure to plastic chemicals and brain structure in young children. We also investigate whether these differences in brain structure are associated with childhood behavior problems, and whether these differences in brain structure may explain the associations between prenatal exposure to plastic chemicals and child behavior problems. In the first study, we look at the effects of bisphenols, and in the second study, we look at the effects of phthalates. These 2 studies provide the first human examinations of early exposure to these plastic chemicals on brain structure. Participants for the first study included 98 mothers and their children recruited from the larger APrON study cohort. Mothers provided a urine sample during the second trimester of pregnancy, from which levels of urinary BPA were quantified. Then, when children were between the ages of 2 to 5 years, they completed a diffusion magnetic resonance imaging, or MRI scan, which allowed us to delineate the microstructure of white matter tracts in the brain. Further, for 56 of these mother-child pairs, mothers completed a checklist of common childhood behavioral problems within 6 months of the MRI scan. We found that exposure to higher levels of prenatal BPA was significantly associated with alterations in brain structure in 2 white matter tracts. Specifically, that higher prenatal BPA was associated with greater mean diffusivity, which provides information about white matter development, with greater mean diffusivity being indicative of less developed white matter microstructure. We found greater mean diffusivity in the splenium, which is shown in green, and associated with mental health problems such as anxiety and depression, and in the right inferior longitudinal fasciculus, or ILF, which is shown in red and is associated with visual processing. Further, we found that these alterations in the splenium were associated with more internalizing problems in children, and that these alterations could potentially explain the association between prenatal exposure to BPA and internalizing problems in young children. Participants for the second study included 76 mothers and their children recruited from the larger APrON study cohort. The methodology for this study is similar to the first. Mothers provided a urine sample during the second trimester of pregnancy, from which levels of urinary phthalate metabolites were quantified. Then, when children were between the ages of 2 to 5 years, they completed a diffusion MRI scan. Lastly, mothers completed a checklist of common childhood behavioral problems within 6 months of the MRI scan. In this study, we found that prenatal exposure to higher levels of phthalates was significantly associated with less developed white matter microstructure in 4 white matter tracts of the brain. The left inferior longitudinal fasciculus, or ILF, which is shown in red and is associated with mental health. The right pyramidal fibers, which are shown in purple and associated with movement coordination and motor function. The right inferior fronto-occipital fasciculus, or IFO, which is shown in yellow and associated with speech and emotional processing, and the left and right uncinate fasciculus, or UF, which is shown in green and associated with language and mental health problems such as depression. Further, for 2 of these white matter tracts, which are marked by 3 asterisks, we found additional associations with child behavior problems. Regarding the pyramidal fibers, we found that higher exposure to prenatal phthalates was associated with less developed microstructure in this white matter tract, and this was associated with more internalizing behaviors in children. Similarly, regarding the IFO, we found that higher exposure to prenatal phthalates was associated with less developed microstructure in this white matter tract, and this was associated with more internalizing and externalizing behaviors in children. In summary, the results of these two studies showed that higher prenatal exposure to plastic chemicals was associated with less developed white matter microstructure in children. Further, we found that less developed white matter microstructure was associated with more behavioral problems in children, and that these microstructural alterations may explain this overall association and thus be a mechanism by which prenatal exposure to plastic chemicals affects behavior in young children.
Hello everyone, I'm Dewey, and I will be talking about the effects of exposure to EDCs, specifically bisphenol and phthalates, on children's neurodevelopment. A growing body of research has reported associations between prenatal and early childhood exposure to these chemicals and children's neurodevelopment. This research has suggested that the effects of bisphenols and phthalates could be different for boys and girls. Specifically, research has reported associations between higher levels of exposure to these chemicals and poorer mental and language development in early childhood and poorer executive function in school-age children. Higher levels of prenatal exposure have also been associated with more behavior problems, specifically externalizing problems in boys and internalizing problems in girls. In addition, higher levels of exposure have also been associated with diagnoses such as ADHD and autism spectrum disorder. However, some studies have not found associations between prenatal exposure and child neural development. These inconsistencies could be due to factors such as sample size, level of prenatal exposure, age of the children at assessment, or socio-demographic characteristics. Our research on the effects of EDCs on children's neurodevelopment included about 500 maternal-child pairs from the APrON study. We measured bisphenols and phthalates in urine samples collected from mothers in the second trimester of pregnancy and at 3 months postpartum. At 2, 3 to 4, and 5 to 7 years of age, the children participated in neurodevelopmental assessments that examined their cognitive, language, executive, and motor functions and their behavior. In the following slides, I will present some of the findings of our research to date. In the first study that I will present, we investigated the associations between maternal, prenatal, and postnatal exposure to BPA and BPS and children's executive function development in the preschool period. So what are executive functions? They include mental processes that enable us to plan, focus attention, remember instructions, and perform multiple tasks. In preschool children, 3 important aspects of executive function are inhibitory self-control, which allows us to inhibit impulsive actions and control emotions, flexibility, which allows us to shift attention and control emotions in response to different demands, and emergent metacognition, which includes the processes of working memory and planning. During early childhood, the development of executive functions is sensitive to environmental factors, and this includes exposure to neurotoxic chemicals such as BPA and BPS. Previous research has reported that prenatal exposure to BPA is associated with difficulties in emotional control and inhibition in preschool children, with larger effects reported in girls. Other studies have reported that prenatal exposure to BPA is associated with difficulties in working memory and planning skills in boys. However, no studies have examined the effect of postnatal exposure to BPA. Further, no studies have examined the effect of other bisphenols such as BPS on executive function development in young children. We used latent change score structural equation modeling to examine if maternal levels of BPA and BPS in the second trimester of pregnancy and at 3 months postpartum were associated with change in executive function from 2 to 4 years of age in children. For inhibitory self-control, the initial model showed that pre- and postnatal BPA and BPS concentrations were not significant predictors of change in inhibitory control. However, the moderation model, which included postnatal BPA and the interaction between postnatal BPA and child sex, showed that these emerged as significant predictors of change in inhibitory control. We examined this interaction further and found that for girls, higher maternal postnatal BPA was significantly related to increasing difficulties in inhibitory control from 2 to 4 years. In boys, higher maternal postnatal BPA was not related to changes in inhibitory self-control. For emergent metacognition, we also found an interaction between postnatal BPA and child sex. Examination of this interaction revealed that for girls, higher maternal postnatal BPA was related to increasing difficulties in emergent metacognition. In boys, this relationship was not significant. Thus, higher maternal postnatal BPA concentrations were associated with increasing difficulties in inhibitory self-control and emergent cognition from age 2 to 4 years in girls only. One route of exposure to BPA postnatally is through breast milk, and previous research has detected BPA in breast milk samples. However, the processes underlying maternal transmission and infant metabolism of BPA via breast milk are unknown. Further research is needed on the effects of early postnatal exposure to these chemicals on children's neurodevelopment. In another study, we investigated the effects of prenatal exposure to phthalates on neurodevelopment, including executive function at 2 years of age. Our preliminary findings have revealed that higher maternal levels of some phthalate metabolites are associated with more problems in inhibitory control, flexibility, and emergent cognition. We also found that higher levels of maternal phthalates were associated with more internalizing and externalizing problems on the child behavior checklist at 2 years of age in both girls and boys. It is notable that different phthalate metabolites were associated with internalizing and externalizing problems. We found few associations between maternal phthalate levels and cognitive, language, and motor outcomes on the Bayley Scales of Mental Development at 2 years when we included all of the children in our sample. However, when we looked at girls and boys separately, we found that higher prenatal concentrations of certain metabolites, specifically those that come from the phthalate DEHP, were associated with lower scores on the cognitive and language subscales of the Bayley in boys only. It is notable that these are the same metabolites that were associated with more externalizing behaviors. Epigenetic changes alter the physical structure of DNA, and mounting evidence in animals and humans suggests that epigenetic changes, such as changes in DNA methylation, may link prenatal exposure to neurodevelopment. Prenatal exposure to BPA in animal models has been associated with variation in DNA methylation in a key neurodevelopmental gene, BDNF. In humans, the effects of bisphenols and phthalates on the developing epigenome are poorly understood. However, one study has reported an association between prenatal exposure and DNA methylation in DRD4, a leading candidate gene associated with ADHD. Maternal nutrients, such as choline and folate, are important for brain development, and animal research and some of our own preliminary data suggest they may modify the effects of endocrine disrupting chemicals on children's neurodevelopment. So we are commencing research that will investigate whether the links between prenatal exposure to endocrine disrupting chemicals and children's neurodevelopment is mediated by DNA methylation variation. We will also examine if these associations are influenced by maternal nutrition, particularly by maternal folate and choline levels. Our findings suggest that efforts are needed to protect women from exposure to BPA and phthalates during the prenatal and postnatal periods. Such efforts may help to reduce exposure of the developing fetus and infants to these chemicals and support healthy neurodevelopment. In addition, our research and that of other investigative teams worldwide strongly supports the need for longitudinal studies that investigate the associations between early exposure to plastic chemicals, children's brain development, and adverse childhood cognitive and behavioral outcomes. I would like to thank the APrON study team, my trainees and research assistants, and of course all the APrON families who have assisted in this study. Without their support and involvement, this research would not be possible. I also acknowledge the support of a number of different granting agencies who have supported this research, including Alberta Innovates Health Solutions, the National Institute of Environmental Health Science, the Canadian Institutes of Health Research, the Alberta Children's Hospital Research Institute, and the Department of Pediatrics.